Mr.
Thank you.
Broyles, I'm going to assume that because you made a big deal out of Filmo being Dr. Filmo O, that you lack the PhD distinction. Is this true?
Yeah, I only have a JD, so
All right, well, that counts.
a little something.
As long as there's a D somewhere.
Yeah,
You are embarking on changing immunotherapy delivery. Would you please tell us about that?
yeah, sure. So, so thank you for opportunity to present to you all. My company, NanoCan Therapeutics, is the worldwide and exclusive license holder to a patented technology that was developed at Harvard's Dana-Farber Cancer Institute. The technology is a delivery platform, and I'll say platform because of its potential varied use cases. The focus of NanoCan is in the oncology space. And if you'd like, I do have just maybe 4 or 5 slides that I could show to explain, or you tell me what works best.
Anything that helps you tell your story works for me.
Okay, great. Let me pull up these slides and just walk you through. Okay, can you all see my screen okay?
We can.
All right, great. So our hope is with our technology, which we hope to get into human trials next year in 2025, we're going to start in hormone-resistant prostate cancer at 3 institutions next year. In short, when you think about cancer care, there are a host of treatments: surgery, radiation, chemo. The biggest breakthrough has been immunotherapy, and then there are more cutting-edge things: targeted therapy, stem cell therapy, and hormone therapy have been big advances. But what we'll talk about today in terms of delivery in the oncology space, so we'll focus in on immunotherapy. And the reason is immunotherapy has been the biggest breakthrough in cancer care. And 2 drugs alone account for, this year, they may reach upwards of $40 billion in revenue. That's Keytruda and Opdivo. We'll get close to 40 billion. They about 34, 35 last year. And even with that sizable market share and reach, those drugs only work for about 15 to 20% of patients. We say 25%, but that's a little, many oncologists have told me that's an overestimate of how effective this is. And in large part, the shortcoming derives from systemic toxicity issues from systemic dosing of the immunotherapy. It gins up your immune system to such an extent that it ends up attacking healthy cells. And so our technology is aimed at addressing this shortcoming, among others. So current standard of care, whether you're talking chemotherapy or immunotherapy, is whole body treatment. And so the patient would have an IV administered, and they would be dosed again systemically, whether immunotherapy or chemotherapy. What we're aiming to do with our technology is to only treat a local area, but generate a systemic effect. And the way we're doing that is as follows. The product itself, I'll just focus you on the left, where we call it a seed. Just colloquially, we'll call this a seed. The seed here is a mixture of a patented combination of materials that have never been assembled together to use as a carrier. And at the top of the seed actually has a hollow core that we load in a small amount of immunotherapy into the seed. And the seed, once implanted, actually biodegrades in the human body. Let me actually just put this slide up and walk you through sort of the method of action of how the technology works.
Eric, do you happen to have a prototype of the seed that you can hold and show us?
It's too small for you to see. I mean, it's the size of a grain. I do, but it's the size of a grain of rice. So on my screen,
Okay.
But yeah, it's literally, think about maybe a little bit bigger than a grain of rice.
Okay.
All right. So
you make an incision and you put this grain of rice near the affected area or on the affected area?
No, we use current standard of care. So let's take prostate cancer, which is our first indication. In prostate cancer, the treatment is brachytherapy, often case, and that's just a needle injecting seeds, radioactive seeds. The practitioners like Phil Moe and my other colleague Paul Nguyen, they'll do the same thing. It'll just be NanoCan's patented seed on the tip of the needle. They'll inject that into the prostate gland area. And if, let's say, for example, if it were pancreatic cancer, and pancreatic cancer, patients have endoscopic procedures to biopsy the pancreas. During that endoscopic procedure, we would insert the seeds for that patient population. So there's a method to all the sort of solid tumors that we're targeting. But with respect to prostate cancer, we'll inject the seed into the patient. The seed starts to biodegrade, which you'll see here on the left-hand side of the screen. It biodegrades, slowly releasing small amounts of immunotherapy in the tumor area. What that release of immunotherapy does is it's attracting a variety of immune cells to the area where the cancer is present. It'll attract dendritic cells, macrophages. All of those cells will start aggregating at the site of the tumor. Because it's a low dose of immunotherapy, when I say low dose, we're talking 1/600th of the amount that you would have dosed with, for example, Keytruda. It's ginned up the immune system, but the immune system's not gone haywire, right? With Keytruda, Keytruda is what you call a checkpoint inhibitor. And that and what that does, your T cells, which are very effective cells at wiping out diseases in the body, let's say, but they're also very aggressive and they can hurt you, right? So your body has a checkpoint naturally built in. And a checkpoint is something that keeps the big fellow calm, right? Keytruda is a checkpoint inhibitor. It actually muzzles the checkpoint and it allows those T cells to just go haywire. And oftentimes, particularly in lung cancer, it wipes out the cancer very effectively, but in other types of cancer has not been as successful, and it can cause damage to healthy cells, and people die from taking Keytruda.
Mm-hmm.
With our product, the immune cells are revved up, but they're not necessarily in all-out war mode yet, right? After about a week of the patient having this seed elute immunotherapy gradually in their body, we then bring the patient back to administer a low dose of radiation therapy. When I say a low dose, again, Phil Moe can tell you in his practice, he may do 40, 50 gray of radiotherapy to a tumor. In our case, we're going to do 8 to 12 gray. And the reason is the reason we're doing it is we're just trying to damage the cancer where the immunotherapy has been released, so that when you damage the cancer cells, it actually generates what's called neoantigens. Those neoantigens is what allows those immune cells that have been aggregating for a week to identify the cancer as being wrong, right? And so now they say, oh, I see you had on a Seattle Seahawks uniform, but you actually have a 3-digit number. You're not number 88, you're number 188. So while you look like you belong, you shouldn't be on our team. That's how cancer grows. It camouflages itself among your healthy cells, but the cancer cells obviously are, they're mutated healthy cells, and they don't work properly. So as you know, cellular biology controls everything in your body, your metabolism, your bowel movement, everything is controlled by cellular biology. So if you have more and more mutated cells that don't properly function, then that's when people die. Well, cancer can do that because they look normal and your immune system doesn't do anything to them. We're actually setting up almost a master classroom in the area of where the cancer is targeted to train your immune cells that have been aggregating on how to identify the cancer. Again, once they're trained with the radiation dose. And I'll say one other thing about the radiation dose. Let me go back one slide. The seed here, one of the components has a number of proprietary components, but one of the components is high Z nanoparticles. And the reason the inventor, who is Professor Dr. Will Enwa, who invented this when he was a professor at Harvard and then Johns Hopkins, he's recently taken over the Cancer Moonshot. He is the program lead for ARPA-H oncology. That just happened in the last month.
Eric, this is a jumping off point. I have an aggregate of questions that are building in my head.
Sure.
Can you help me understand where you are as a company? For example,
Yep.
NanoCan, how old is it? When did you join it? How many employees do you have today? How close are we to any kind of clinical trial? Can you give us?
Yeah, let me finish this. Let me finish telling you about the technology, and I'll take 1 minute on that, and then I'll give you all that background. But these seeds have high Z nanoparticles. which under Einstein's photoelectric effect amplifies this lower dose of radiation. So you're not giving as much radiation, which can also be toxic, by the way. Yet at the site of cancer, it's killing more cancer locally, right? So that's another part of the sort of the ingenuity of what this inventor did. In terms of the company, where we are is we have, I founded the company 4.5 years ago, and we have, I don't know, 10 or so employees inside the company. We manufacturing.
Full-time,
I'm sorry?
full-time, W-2 employee,
More than full-time, but yeah, I mean,
overtime,
well,
got it. And you mentioned you have a JD, so you come from a legal background. What medical background do you have that led you to jumpstart this effort?
yeah, so about 12, I practiced law for 10 years and then I went off to become an entrepreneur. So I helped build a very successful company in the professional service space. And then I invested first in a pharmaceutical packaging company, which is basic sort of science. But in my getting in that ecosystem, I just went up the value chain. I launched a transdermal patch company. I'm the creator of the first ever transdermal patch that can prove delivery of vitamin D through the skin. I did that in partnership with King's College in London. We just completed our clinical trials. I've been working on that for 7 years. It's first ever time you can prove D delivery. That's clinical trial. I ran, I set up, I funded the trial along with my partners.
You're
And
a BFD.
what's that?
You're a big fucking deal.
Oh, no, no. But that, in fact,
you also hit a couple of notes because Jan is a packaging engineer and you said packaging and you come from a legal background. That's Mark's background. So you're hitting all the notes.
Yeah, so Jan, my partner, Jan, was a gentleman by the name of Sid Lewis, who used to own International Labs. Sid launched Walmart's $4 drug plan. He came up with the compliance pack that allowed Walmart to economically package drugs in a compliance pack, which looks like birth control packaging, right? And so after he sold his company, he formed a company with me in 2011, and that's how I kind of got in this space. And then through attending various conferences and talking to pharma companies, I just got deeper in. So the transdermal patch was our first foray.
Yeah, but I didn't like you calling it basic packaging. That's what gets so many people in trouble. And I'm seeing a lot of cool things that can happen to this seed.
Yeah, there's nothing basic about packaging. I just was talking sort of it's very complex, and they have to comply with same GMP requirements that you do in making a drug. Yeah. So certainly not to downgrade that industry at all in any way, because it's very complex as well. So with that said, the inventor of this product heard about what my team invented in London and asked me to meet him in Boston. This is 5.5 years ago. And he said, how'd you guys come up with that? I think you can do something with skin cancer with a patch. And I think you guys developed a pretty cool technology. And that's how I got involved. He said, you know what? I'd like for you to be my partner and launch this, to commercialize this technology. That was 5 years ago. We set up a company, NanoCan, which we are manufacturing at a contract development manufacturing organization now. We've done an array of preclinical studies. We have one more IND enabling study to do in large animals. We're going to do that in dogs. We're actually toying around with switching to monkeys because monkeys have now started to become available. We thought we're probably 1.5 years behind, right? Anybody who's done pharma, you hit snags, and we've hit those because it's a very complex product. It's very, very complex.
I'm surprised to hear that you say that you're in manufacturing, which suggests a scale of production that I don't understand, given that you're free.
CDMO. There's a class of manufacturing and drug development. You go from a tech transfer from either an academic lab or even if you're inside to a contract development manufacturing organization. Those entities typically target preclinical and clinical trial stage manufacturing, right? So small scale, just enough that you can dose your animals and dose a small cadre of humans. But with that said, from day one, I've been thinking about, if this works, how do we commercialize it? And so we have started down the path of, okay, let's assume we're successful in clinical trials. Can we actually commercialize it? And I actually think we're pretty good ways to figuring that out as well.
Let me use that as a jumping off point to introduce you to Nick Anderson, who is one of the smartest and most delightful people I know, and he also happens to know a thing or two about health economics and reimbursement. You may find his knowledge helpful. Nick, from what you've heard today, how would this be presented to a value analysis committee in a way that you think he'd be successful?
Yeah, thanks, Joe. Eric, this is awesome. And sorry, I've been multitasking as I have to with
his 6 children, and he works at Boston Scientific, and he's always in the CEO's office. So he's a busy guy.
Oh, thank you. Thank you. No, Eric, this is awesome. And I'm glad that you made the connection back to brachytherapy. And maybe that's how a hospital would look at this, is to say, we already know about brachytherapy and you're just a new one. And that always hurts your feelings, because it's like, I'm not just another one. Like, I'm better, but you go, no, I am just another one. I'll let you believe that if it means you're going to buy $1 million worth of this, you know? So they already have an idea of what it is to implant a seed into tissue, and you're just another seed. So you tell the hospital, like, yeah, we're just a new and improved brachytherapy. And then you can go tell the doctors, like, you know, a different story if you want, but both stories being true and accurate, but it's just what interests them versus them. I think this is fantastic. It's a really cool solution to solve the overdosing of systemic chemo issue. I think you're onto something. This is going to be awesome. And yeah, I do reimbursement, health economics. How do you get hospitals and insurance companies to say yes? And anyway, you're nailing it. This is a fantastic technology for the little bit that we've heard about it this morning.
Oh,
so
you.
Thanks for taking the time. This is awesome.
So you ride on the fact that, yes, it's just another seed to say, yes, those CPT codes work just great for me.
Yeah.
Yeah, when you're saving lives in cancer care, it's a different discussion though with the payers too. They're not. I get it. If we were another blood pressure medicine, then yeah, you got to jump through a lot of hoops. If we get efficacy in pancreatic cancer, it doesn't matter.
Mhm.
It almost doesn't matter.
Now, I'm not as familiar, of course. What kind of efficacy levels do we see today in pancreatic cancer? Because I'm not aware of any, really.
Yeah, so my collaborators developed the standard of care. So Amal Narang at Johns Hopkins, he now has the standard of care for pancreatic cancer treatment. It's not very great. You maybe 6 to 8 months under his treatment you can get, but you know, it's a very aggressive cancer. Amal has convinced one of the large pharma companies that if NanoCan can prove that little seed is safe, that they should partner with us after we do our phase 1 in prostate to do a phase 1B, to just do a small safety in pancreatic, and then to partner with this large pharma company to test this in pancreatic cancer as well.
Of course, this is a speculative question, but if the standard of care buys you maybe 6 to 8 months, what do you hope, believe, aspire to achieve with your formulation and your platform?
Yeah, so if you look at our Kaplan-Meier curve for our pancreatic model, we basically got 5 to 7 times the length. Let me actually pull back up the document. I'll just actually show that. We got 5 to 7 times the time. So perhaps we could get the person another 18 months, 2 years maybe, which, you know, that my dad died of lung cancer. If somebody told me they'd give me another year with him, I would take it.
Yeah, no, I hear you. That seems like a pretty convincing yes to me.
Yeah, this is an example. So this is our rodent model, and we've done all sorts of rodent models at Hart. We've been doing this for 7 years, but we only treated the primary tumor, which is represented by the green shade, right? We didn't treat the red, which represents metastatic spread. And through this process that I've described to you of the immunotherapy attracting immune cells, we then damage the cancer with a low dose of radiation therapy, generating neoantigens, which identify the cancer as being wrong. Now your healthy immune cells, they have a roadmap. They have a DNA toolkit to figure out, okay, now I know what I'm looking for, right? This 3-digit bandit with blue eyes and a gray helmet and whatever else, whatever other unique identifiers you have. Those cells go out through the lymphatic system and train those T cells that I talked about earlier, that Merck's Keytruda gens up, right? They go out and train them. They say, hey, here's how you recognize the cancer. They got a 3-digit code and they got this other thing. And then those cells get involved, and you see the result here where it wipes out the cancer metastatic, even though we only treated one local area. And I'll say one other point about this. This is a Kaplan-Meier curve I mentioned to you in terms of how much longer. So pancreatic cancer, you can see how much longer we extended, extended life. You at least got double the time. For pancreatic, for prostate, you got more. But the important thing about this study is when we reinfected these rodents with cancer, it didn't take, which demonstrated Professor Enwa's thesis is that you could develop sustained memory in the cells for that type of cancer. So none of our rodents died from cancer. They died from other causes. They were vaccinated from subsequent cancer.
Could you go back to the previous slide? If
Sure.
you'll indulge me, one of my, again, indulge me, gifts as a moderator is I have no problem having everyone on the call understand exactly how dense I am and how difficult things are. So I'm going to ask you like you're talking to a child. How many rodents are represented on this slide? Because it looks like, I mean, to me, the same one, of course, but it's not. You have four different rodents that are here.
And multiple cohorts of 4 different rodents, right?
Okay.
It's not
So
like we just took
that's 4 for
4
now.
rats, put cancer in them. So these studies, there could be 32, there could be 50,
Okay.
groups
So they
of 4.
represent 4 cohorts.
Yes.
The one on the left is we did nothing.
Yep. Control group. Yep.
The one in the second is we radiated and
Got radiation. Yep.
okay. The third is immunotherapy, and it helped the left a great deal, but not
Quite a
all
bit.
the way. And on the right, it even got worse, it seems. Well, not worse than the control.
Yeah. It's about,
Using
yeah.
more treatment, it got rid of everything.
I have a
And-
question on the immunotherapy only picture. First of all, you're way out of my league because I'm just an engineer, electrical engineer. But I thought you just said that with ordinary immunotherapy, you flood the patient with the same therapy. And I would suspect then that with immunotherapy only, you cannot just treat the left tumor. Or did I got
No,
the treatment wrong?
No, you're right. You're absolutely right. You're absolutely right. The point of immunotherapy is it's everywhere in the body, right? So if it's effective, you would think it would get the local tumor and the metastatic spread, right? Because it's circulating. The immunotherapy is going throughout the body, throughout the bloodstream, but it doesn't, and it doesn't tend to work on metastatic spread. Again, works Keytruda and Opdivo both work very well in lung cancer, and they work in certain blood cancers, and they've had some efficacy in breast cancer. But 80% of patients don't. It doesn't work.
So in this case, only the left tumor treated is a bit, how do you say it? It's where you applied it or something?
Yeah, well, remember, so let's say this right here with immunotherapy, they got an IV and they shot it through the blood. With us, we implanted our little seed inside of that green tumor, right? So that's the difference. This seed right here got injected into that green tumor. So it's not circulating in the bloodstream, right?
Okay, it's not going to wander around in your body?
What's that?
It's not going to wander around?
Well, after it's been trained, yeah, after the immune cells are going to be trained in terms of now, and again, so 2 things. We're using 1/600th of the amount. So if it did roam around, so what? It's not, it's not generating again the effect of of a Keytruda, again, which is a checkpoint inhibitor. It's muzzling the thing in your body that keeps your T cells in check, right? So that they don't go out damaging everything in your body. Keytruda is a temporary blockage of those those checkpoints so that your T cells can get revved up and attack everything. And that's exactly what happens with Keytruda. With ours, again, we're inserting the seed into the tumor 1/600th of the amount that, by the way, is slowly releasing over several days. So even that 1/600, let's say you released it all on day 1, you're talking about a small amount going out over anywhere from 8 to 20 days. We'll figure this out, further in our when we do our PK study in monkey or dogs, what the timing is. But let's say it's going to be somewhere from 7 to 20 days that it's slowly releasing very, very small amounts. So
Only one in one tumor, or multiple in one tumor, or multiple in multiple tumors?
one tumor, yeah, one. So as represented here in the animal model, we only treated one tumor. Because again, we're training your immune cells how to recognize your cancer. And I should make that clear too. It's personalized to you. So not only is it precision medicine, it is personalized medicine because your immune cells are getting a master class on how your cancer mutates, how it impacts. That's one of the problems with treating cancer. There are billions of iterations of the, of the cancer cells in the patient's body. This is where
Those high-seas particles are quite nifty.
Yeah. Eric, since I, when I do due diligence, also ask questions that are basic English, do you want me to take a stab at explaining this the way I did to our investment group?
It's up to you, Maren. I don't mind. It's your group. Yeah.
So let me try to...
Before you do, Maren, I have another question that's, I think, pretty basic. If you go back to the rat slide, please.
Yeah, sure.
As I understand it, a metastatic spread is the same cancer moving somewhere else. So
Correct.
why would it treat, why would there be a difference between the, I see, you're saying that it handles the metastatic spread because it recognizes the cancer everywhere despite, because I thought the major point you were making is the seed is localized. So if I have cancer here and here, and you put the seed here, I would think that your point of difference about localization is I'm fighting it here. So this spread,
Can I try, Eric?
okay, Maren.
You can give it a try.
Let me try it. So this is the way I understand it. You have this little seed with a small amount of the very toxic chemical that's going to treat the cancer.
Yes.
You put that in the localized tumor that you've identified.
Yes.
You have a local effect of killing the tumor there.
Yes.
But you also have a more general immunotype therapy effect of exposing the cancer cells to the standard, just what I'm looking for, standard cells in your body that fight things off, like T cells and things like that. And so you train those T cells locally because they're seeing the exploded dead cancer cells to recognize that as a foreign body or an intruder in your body. So now that you've trained those cells locally, now they go and they distribute themselves through your body, and they're able to identify wherever that tumor is happening in your body. So
they actually go out and train other immune, like your T cells are not getting trained during this process, but the T cells get trained when these other, your body has a whole host of immune cells, right? Dendritic cells, which are really smart. They're like Navy SEALs. T cells, they're like big bomber jets, right? Macrophages, which aren't very smart, but if you train them, they can be very effective, right? At what once you sort of point them in the right direction, so your body has a host of immune cells that all are getting, so those cells that are at the local area, they're getting trained. They go out through the lymphatic system and train your T cells. The T cells are out here, right, where this metastatic spread is. Now they know I'm looking for somebody with a 3-digit, in a Seattle Seahawks uniform, but they have 3 digits. And that's what they did. They found all the 3-digit or whatever the unique marker. It may be 50 items that are unique about that cancer, but they've been trained on how to recognize all 50 of those markers, right?
I tried an analogy, see if this works.
Yeah.
Your seed is general pattern, and you know where there are a lot of soldiers, tumor. You insert general pattern where there are the most soldiers, and what he does while he's there is educates those soldiers about the bad man, and then the soldiers go out throughout the body and find all the bad men they can and kill them.
Yeah, I've used a military analogy. I've used the police analogy I use is this. Keytruda's drug is let's say law enforcement, right? It's law enforcement coming up and they say, oh my goodness, officer down, right? A police officer has been shot. That's going to rev up the entire police department. They're just going to come in guns a-blazing looking for the person who shot one of their comrades. What our technology is doing is saying, hey, officer down, but by the way, we have a mugshot, we have a fingerprint, we have a DNA sample, we have their disguise kit. We actually know what disguises they're going to put on, right? So while everybody is still revved up, they're much more informed about what they're looking for, right? They're still psyched because one of their comrades got shot, but they got a mugshot, a fingerprint, a DNA, and their toolkit in terms of how they disguise themselves.
That's that helped a great deal. So thank you between the two of you. I now kind of understand. I'm also thinking that I'm thinking that cancer cells mutate into new strands, and that's why, even though I killed everyone who had that fingerprint, I didn't kill his friends that are kind of offshoots. And there's just so many generals that you can insert to fight all the different people. That's where, because the concept of, and then we find all the bad men and we kill them.
Mhm.
Okay, then why am I only lasting 18 months if I killed all the bad men? No, there are new bad men coming. Okay, well then I'll insert another guy. I mean,
Well,
how
but
come-
that's why I said to give you the disguise kit. We say, oh, by the way, the bad man has a toolkit of disguises. He's got a fake mustache. He's
Mhm.
got
I get
bulky eyelashes. He's got a blonde wig,
Well,
right?
we're going to kill anyone who's a bad man because you taught me all of his characteristics. But is it that the bad man mutates into something that is not characteristic? I'm wondering, again, as silly as the question is, but taking us down this analogy, if I've trained every soldier to kill every bad man, how come the patient dies after 18 months? Why
How
hasn't the
come what
cancer gone
patient, how
away?
come what patient dies after 18 months? What patient are you talking about?
What you
One
said
that's had
that
this treatment?
That he's talking about so I think what Eric
the
said,
standard of care gives you another 6 to 8 months of life. Your care would give me 18 months of life. Why not
Or a cure,
years?
or a cure? No, it could. We don't know. That's why you do clinical trials. This could cure it. We don't know. I just don't like using that word.
Yeah,
And that's
okay.
not my objective, right? The last 2 drugs in pancreatic cancer that got approved, one extended life by 3 months, one extended life by 9 days.
Okay. Is that where you were going, Maureen?
yeah, the endpoint for cancer drugs, since it's awfully difficult to show that something is actually cured,
Mhm,
and it's a long time,
mhm.
so the approval criteria is typically extending life.
It is. That's it. That's the
Okay.
that's
Because
the
otherwise
milestone.
you'll never get these drugs out in the market because they're trying to hit an endpoint that's really, really difficult to show.
Mhm.
And what I am thinking of it is more like a vaccination, the seed, and you're using the radiation to break down the outsides of the drug can actually work properly with the T cells. Am I thinking wrong?
Yeah, only in the sense that if you think vaccination is, it operates as a vaccination, but it's like a ex post facto vaccination, right? It's
Yeah,
after the fact, right?
because you're using existing drugs, you're putting the gel around it. So there's a lot of other drugs once you figure out if this works right and doesn't hurt anybody that you're going to be able to use this with.
That's, you hit the point, Jan, which
Well-
also gets at Joe's point, right? About how cancer mutates. Joe, and Jan, we can load this seed. We can load this seed with Keytruda. We could load it with Opdivo. We could load it with chemotherapy, right?
See, to me, your gel is the package.
Yeah, the seed, the delivery, the delivery, all of the patented materials that we use to carry the actual active drug. You're correct. That is the platform. And by the way, we could use this in non-oncology indications as well.
Jan, I think there's one little nuance. The cells are broken down to be able to train the immune system, not to make the drug more effective. So the drug helps breaks the cells down. The fact that the cells are exposed cause the immune system to react, and that's what happens. That's how it goes forward.
And I think that's a good point. And I would actually say that is if you looked at the competitive landscape, you guys might see, well, Eric, there are a bunch of companies out there doing slow drug release or doing things that look similar to yours. Marin hit on a key point, which is they are trying to use their drug to wipe out the cancer. We are leveraging the drug and our system to train your body's immune system on how to effectively wipe out the cancer. That's the difference. It's a different method of action. We're not using the drug. We're using the drug to kill a little bit of the cancer, but only to the extent that you can do an autopsy on that dead cancer so that everybody's like, they have what I'll call recognition. And recognition is key. Oh, it's saying my battery is running low. Let me just plug up.
And that's why I think of it, my mind to wrap around is sort of like a vaccination, because that's what you're trying to do with the vaccination is train your body to recognize whatever that disease stuff is, but it's through your whole body for it. And you're going a little tiny piece to make it work better for the whole body. little bit different, but similar to me. I'm just a packaging engineer.
Mhm. You're more than just a packaging engineer.
Well-
Jan. Now, what's Filmo's specialty?
So he may be with a patient now, so I'll brag for Filmo. So Filmo is a MD, PhD. He's a radiation oncologist, so he treats patients. He's also a professor at University of North Carolina, Chapel Hill, their medical school for radiation oncology. But his specialty was on nanoparticles in the tumor microenvironment. So he is a whiz at this and knows this almost as well as the inventor, who is, probably on a little different level, which is why the White House brought him in to figure out. And I'll just tell you why the White House brought Professor NY in. They said, you have the thinking to almost democratize access to immunotherapy, right? Because immunotherapy, Keytruda is expensive. It could be $150,000 for your treatment. And what the Biden administration, in the short time that they have left, they figured out, hey, you come in, work with ARPA-H, and figure out, you've thought about this in a way that would make immunotherapy broadly accessible, even to low and moderate income people, and lowering the cost of healthcare in the U.S. system. And so that's so he's really got interesting ideas on this, among others. So Filmo, again, radiation oncologist, PhD in immunology, treats cancer patients, but also likes to tinker around with the actual product itself. And then here, I'll just pull up the team right quick. I'll do this in two.
Meanwhile, Mark, you had some thoughts. I'll remind you, Mark is your fellow JD.
Yeah, I mean, I just had a quick question. I mean, the alternative is trying to train the immune system outside of the body and then injected the result of that training. How does that compare with this approach from an efficacy point of view?
Yeah, what you're talking about is like when you see sort of these designers, certain stem cell types of therapies and things like that.
Yes.
Right. So a couple of different ways. Number one, those are very expensive, right? You're talking $1 million for the patients you're talking $1 million to have that done. And they're also very painful and time consuming, right? So those are just some very practical considerations that you would just have to take into account. How many people?
No, I realize this is obviously much more cost effective and is a much better solution. I was just curious about the efficacy of the one approach versus the other approach.
So I'd look if Filmo, if he were here's my sort of response to that. Do you, just from a pure logical perspective, training something inside the body where all the action is going on versus training it in a petri dish outside the body, which one do you think will likely have a better sense of all the ways things might mutate and things like that? I think having the training occur in the body is another key part of this innovation, right? Where you're actually in the environment that you need to operate in. When you take things outside of the body for these sort of designer drugs, which is why some work, and some have been, effective, but a lot have not. And it's a very complex process.
Yeah, but there's targeting disadvantages too. So I'm wondering, I just think it needs to be tested, right?
Yeah, that's right. We'll know more once we get into humans next year. We'll know more. But our thesis is that this is the training your cells in your body is much more effective than trying to train them outside your body.
Thank you.
Yeah, you're welcome. Oh, yeah, I was gonna just show you guys some of the team members here. So this is the team that's working on it. So you've met me. Paul Nguyen actually hired the inventor at Harvard 20-something years ago. Paul is the vice chair of radiation oncology at Dana-Farber. He's the #1 prostate cancer doctor in the United States. Mm-hmm. Adam Schilling is our chief development officer. He began his career as a bench scientist at Pfizer in toxicology. He's a PhD in toxicology, but then he spent the last 18 years just on regulatory affairs. So he's been running clinical trials for companies, and he's taken 2 cancer products from preclinical to registration in that 18-year period. And Filmo, I talked to you about, and Dave Krasny is our CFO. And then we have a scientific advisory board. Paul Wallner, up until 2 years ago, every radiation oncologist in the U.S. has his name on their diploma. He certified you to practice radiation oncology in the United States. He was the chair of the American Board of Radiology, and he retired again 2 years ago. Elder Granger is a oncologist as well, a blood cancer specialist though. He ran TRICARE for our U.S. military, board member of Cigna Health. Brian Rennie runs Vanderbilt University's clinical trial group, but he was at Cleveland Clinic for many years. And then Jonathan Yang, who was at Memorial Sloan Kettering. He actually now is the director of research at the Langone Cancer Center at NYU. And these guys have all been involved with me for years. Jonathan and Brian for 4.5 years, Elder and Paul for three and a half years on our team. And we have a host of other collaborators.
Can you tell me how much has been raised so far, and are you in a seed round or?
Yeah, so we did a pre-seed round, was just myself and one of my early co-founders who unfortunately passed himself from a heart issue. We were college classmates.
Oh.
Greg Wolf. Yeah, so we Greg and I initially put in $1 million between the two of us just to kick the tires on this to see what we could get going. We ended up raising in our pre-seed round about 2.7 million for pre-seed. We did a round of convertible notes at 2.6 million on top of our 2.7. We've gotten 3 million in NIH funding. So again, you're approaching kind of 10 million of funding. And then we're in the middle of, we have a round open right now where we're raising 4 million. We've raised most of that. I think we've raised 3.4 or 3.5 of the $4 million. I think we have 5 or 6, maybe 600,000 left to raise on that round. And we may have to up the round just because we've hit delays. And it's a discussion I'll have with my investors as we.
How many investor groups or shareholders do you have at this point?
So I'll give you the big ones are, so Marnes Group, Alliance of Angels, they're a 6-figure investor. Park City Angels is a large investor. St. Louis Arch Angels is an investor. Silicon Valley Angel Groups are investors. Prevail Partners is a venture capital fund. They led this $4 million round. They're an investor. What's the other? Life Force Capital was a very early investor. Life Force Capital is an investor, and the rest have been sort of wealthy individuals and family offices, including Paul Schimmel, whose work led to the mapping of the human genome. Right. So Paul is the second most famous professor at MIT, with Bob Langer being first. Who I've met Bob Langer through Paul on this product, by the way. When I hit snags a year ago, I went up to see Bob to get some advice from him. And what happened was one of his former students who works with MD Anderson down in Houston has been consulting with us for close to a year now and helping us really solve some of the development challenges. But that's a sense of the type of folks. I mean, it's just well-heeled, smart. It's all smart money. CEO of Citigroup Europe is an investor. Fairly sizable. So, smart, former owner of the Denver Nuggets is an investor. So, smart money.
What do you surely they ask you for some kind of timeline or expected, recognizing delays and the like. What do you tell them?
My goal was to be in trials this year. Was to be in trials this year, but candidly, you can't get by the FDA though, right? Like, I just have to tell you, it's like one issue, right? That I think we have it worked out, but I've been working on it for over a year, right?
What is that? Regrettably, my friend Michelle dropped off the line because we're approaching the top of the hour. She's my FDA expert. What is the issue? I'm recording this, so perhaps she can...
Yeah, so I'll just, at a high level, it's too complex to go in on this call, but at a high level, before you can file what's called an IND, you need to have well-characterized analytical method development so that the FDA can measure how is the drug released into the body, can you recover it, and can you show its activity after it's been recovered, right? So release, recovery, and activity assay. That's very critical. Remember, we're dealing with... So let's take, for example, Keytruda, right? And let's say they're putting, and I'm making up a number, a pint of Keytruda in your body. Yeah. The FDA wants to see, okay, how much of that pint can you recover in the plasma of the subject, right? And then after you recover it, was it active? Did it work? They need to know, does it work? And so that's why I said it's too complicated to explain, right? Because
and here I'm just going to play the dumb card. What do you mean recover? Like if you do a blood donation, you're going to measure how many what percent of the blood you've extracted has the drug in
No,
it?
Joe, I think I can help.
Please.
This sort of thing goes back over 100 years. In World War I, they had just recently discovered antibiotics.
Okay.
You can actually recover the antibiotics from the urine of the patient who's been dosed. And it was so expensive back then that they would do that and be able to use the antibiotic again to cure other patients. So there's an element of
purify the antibiotic out of the urine to reuse it, recycled
Yep.
antibiotics.
Yeah,
So I mean that's but
saying
one
no.
example of where they would literally recover
You have a
and
different understanding
determine
here?
its activity.
so let me just make it real simple. The FDA wants to know what's being put in humans' bodies, right?
Right.
They want to know that's it. At a simple level, the FDA wants to know, what are you putting in a human,
Yeah,
and is it active? Do you have a way of showing me, the FDA,
That it's
that what
working.
you're putting inside of a person is active? And I'm saying that's what's been challenging for us, because we're using micro doses, right, of something. So, but we still, the FDA doesn't give us a pass, right? Because we're saying, hey, we're using so little, it shouldn't matter to you. They don't care. They want us to measure it, and they want us to show us its activity. And that's been challenging for us to figure out. I think we're close to having it solved.
Eric, I mean, again, so going away from that example, in fact, what you're doing, one of my previous companies was involved in phage therapy, which again ties into the antibiotic world, but scales. And the problem is, I mean, for you, you're actually your active is actually just promoting the development of the immune system.
Exactly.
So you actually aren't really wanting to show that you can recover and show that that little bit that you put in is active. Shouldn't you want to be showing that you're actually increasing the immune response system?
We do, but the FDA wants to know what else. Show me. Yeah, that's great, Eric, that it works, but show me what was working.
How do we know you did that? Yeah, exactly. So
That's a challenge. But there are all sorts of tests, and we through all the experts we've talked to. So that's an example. And then manufacturing the seed, it's very hard. It's a complex thing. It's not like this is a very, very complex product. Fortunately, through just a series of fortunate occurrences. I got former senior J&J manufacturing executive who's come in recently and kicked the tires on everything, is really helping us to solve a lot of these problems. A guy who's retired from J&J was like a big biologics guy for them, so he really understands working with these type of large molecules and nanoparticles, and he's really been helpful to us. So but we're going close. So yeah, if I had to, what my investors know is we're about 1.5 years behind, right, of what I told them when they were putting in.
What did you tell them?
Well, we wanted to file the IND
Right.
at the end of 2022, right?
Mhm.
Because that's when we wanted. And so it's gonna look like filing the IND. I don't think we can get it even this year. It's possible. It's not 100% out of question, but it's unlikely.
I'm confident that your network in this field vastly outstrips mine, but if I were a magician or wizard of some kind, how would you finish this sentence? Yeah, Joe, if you could put me in touch with someone who that would really alter our path. Who do you need?
Yeah, at this point, we kind of have all those people now. So there's not, I mean, a year ago, I could have given you 4 or 5 names, right, of types of people. But at this point, we kind of have, that's why we're solving the problem, is because we have the people, and we're pretty much, the problem is it's very close to being solved.
Mm-hmm. Okay. Well, it was something really talking 45 minutes nonstop about Filmo and also talking about the drug treatment. So Filmo, I'm sorry you missed it. It's all in the recording, a lot of trash talking, about your contributions or lack thereof. And,
Every time I talk to
apologize,
guys in the golf course, I'm like, I picked the wrong profession. Even as a lawyer, I didn't golf as much as him, and I think he's a scratch golfer too, which is amazing.
I apologize. I'm covering for 2 doctors today, so kind of got pulled in different directions.
If you're a really good golfer, you too can run for president. I think that's how I understand.
I got a better swing than Trump for sure. This is painful to watch.
Oh, I don't want to hear that kind of trash talking on my channel. I don't need to be docked by, the Secret Service or something.
Right now they're watching us.
Yeah, I'm going to edit that out. Rick, what a pleasure. Thank you for sharing your work with us. I I wish I could say something like, and because of this call, we're going to get you the things that you need, but you're there. in terms
Money,
of
I guess we still need some money. So you can talk to Maren about that or whatever. But no, so we're working through. But thank you. This is great. Very good questions, by the way. Very, very helpful questions.
We are few but mighty, and our disciplines are quite varied. So it makes it a real pleasure to meet with these folks every Friday morning. Eric, Phil, thank you so much.
Eric, if you need help in the future, you now have a very good contact.
I get it. I get it.
Thank you, Maren, for connecting us all, and have a great week, everyone. I'll see you this time next week.
Thank you.
Thank you
Thank you
very
all.
much.
Appreciate your
Thank
time.
you.
Thanks.
All right.
Take
Bye now.
care, folks. Bye-bye.
Cheers.
Bye.